Finally, I want to discuss with you today recent breakthroughs that I feel that became very, very, very important recently to enable us really, eventually, I think it's like a tour de force really, in an attempt to understand neurons as computing device beyond ideas. Because all that I showed you before were theoretical ideas. I did not really know how a particular neuron computes the direction of motion in the visual cortex. These were ideas. Now, what do I need to know in order to make these ideas concrete and really solve the issue? How does this neuron compute direction of no motion, for example? So I want to highlight basically two really experimental breakthroughs that really brings us close to be able to answer these kind of questions experimentally. One work comes from Arthur Connor's lab in Munich, whereby he developed the optical system, the two-photon microscope system that I mentioned already before, to the level that enables us to ask really, at the level of single neuron, questions like, how does the neuron compute direction of motion? So here is a very fast experiment. So here is a mouse looking at the world. Here is the world. And the world, these lines on the screen are moving in this direction or in this direction and so on. And he records from a particular cell. So this is the cell he was recording from in layer 2, 3 of the cortex. The visual cortex of the mouse, V1, records from this cell with an electrode, showing the different directions to the living mouse. So the line was moving this direction, recording from the cell body. Then you see ta-ta-ta-ta-ta-ta, the cell fires. In this direction of moving bars, much less spikes. In this direction, from here to here, more spikes, less spikes, and so on. You see that this is a directional selective neuron. Because in some directions, particularly this one, from bottom to up, top, it fires a lot the cell, it fires a lot, less, no, less, and so on. Directional selective cell. Okay? So if I sum what I just said, I said that this particular cell is oriented. It responds to this direction. You can see that this polar plot, there are a lot of spikes when the movement is in this direction. This is this case. And there are also spikes when the movement is in this direction, which is this case. Okay, so this is the computation that the cell performs, is computing the direction. In this case, the cell likes this direction and this direction. But how does it do it? What do I need to know? How does it do it? So Arthur Connor extended these techniques of two-photon microscope to be able not only to look at the output of this particular cell, but also at the orientation selectivity of the inputs, of the synaptic inputs to this particular cell. So this is a summary from his paper in Nature from 3 years ago. And here is the result. The result is that if you zoom in into this input synapse, into this EPSP, you see that this particular input synapse is sensitive to this direction, meaning that this synapse is active when there is line moving or bar moving in this direction. This other synapse, let's say synapse number 4 here, likes these two directions. You can see it here. Synapse number 4, the EPSP, using light, calcium sensitive dyes locally here, or here in this case synapse number 4, you can see that synapse number 4 likes to respond to this direction and also here to this direction. So this is synapse number 4 that likes to give to respond to this orientation and to this orientation and so on. So we are now at a really miraculous time where not only you can record the output of this cell, which is a summation of all its inputs, but also you can record from the input sensitivity itself what direction this synapse is sensitive to, or this or this. So for example, you see in this case, something we did not know really, that this particular cell, the layer 2-3 pyramidal cell in the mouse visual cortex, V1, all orientation are presented on the dendrites. So the dendrites experience as an input all the orientation, all the synapses, the different synapses code for different orientation onto the dendritic tree. But the output is oriented to a particular orientation. Like the input that is sensitive to particular orientation, also the output of this cell is sensitive to a particular orientation. How does this dendritic tree transform the sensitivity of the dendritic tree, so to speak, to many orientations, to making an output representing a particular orientation? Unsolved as yet, we don't know, but some computation is performed on the dendrites. For example, it could be, we don't know, that most of the synapses, it has many, many synapses here, not only 20, but 5,000. Maybe if you just count all the orientation selectivity of the input synapses, you found that most of them are 90 degrees oriented, the inputs. So the cell just is a follower of the majority, just majority. It's a democratic neuron, just counts the majority of orientations and decides that the majority dictates my orientation. Maybe there is another computation being performed on the dendrite. This will be solved, I'm sure, within a year or two. That's for sure. That's one interesting and important new technology that enables us not only to record the output of a cell in vivo, in the behaving, in the full in vivo situation, but also the input to this cell, and some computation is being performed by the dendrites, generating a particular directional selective output. The other system I want to discuss is direction selectivity in retina ganglion cells. Very fast about the retina. So the retina is built from several layers. The most important ones are marked here. So the receptor layers is the layer of neurons receiving light. So light comes in, absorbed, the photons are absorbed by the receptors. And then there are several stages, the bipolar cells, which is the next stage, receiving input from the receptors. And then the next stage, the amacrine cells here. And then the next stage, the ganglion cells. And these ganglion cells have these axons, the optic nerve. So the output of the retina is here. The input to the retina light is here. And there is a network in between. And this network eventually computes something, because already in the retina, if you record from some of the ganglion cells, you see that these cells are directional selective. Here is an example. So this is a recording from a ganglion cells in the retina. And you see that for some direction of motion, there are spikes. For some other direction of motions, 80 degrees, 135 degrees, there are no spikes. So this is a tuned cell that tends to fire only in the retina already, only when there are this direction of motion, 225 degrees or 270, and no others. So I'm saying that already the retina, before the cortex, before Hubel and Wiesel, before the cortex, there are already cells that are directional selective. Even when you cut it from and you take the retina out, the computation is implemented at the level of this local network, at the retina. How? How? So there were many ideas along the years. One of the most fundamental one is what is called the Reichardt detector. The idea is a theoretical idea. It comes with many ingredients, but the general idea that there is some asymmetry in the system, whereby in one direction there is more inhibition coming at some location than in the other direction. So some asymmetry within the network causes one direction to involve less inhibition compared to the other direction somehow. And this may explain the fact that in some direction, in the null direction, you will not get an output. In the preferred direction, you will get an output. And particularly for the retina, this was a possibility suggested. And here is just an example. So if you move a light in this direction, these are the receptors. The photons are being absorbed. And if there is some kind of asymmetry, so this is the ganglion cell, and if there is along the way asymmetry in terms of location of inhibition compared to excitation, then this system will generate direction selectivity, because if lights move this direction, this receptor will activate this inhibitory cell, amacrine cell in the retina, and this inhibitory cell will be activated first, just because you move here and this cell is activated. Bipolar cells activate this amacrine inhibitory cell. This inhibition impinges on the ganglion cell, so you inhibit this cell, and then when next you come here, the excitatory path is already inhibited by this cell that was previously activated, yes, and then there is no output. But if you move the other direction, you first activate excitation, and then later on you activate the inhibition. This inhibition comes too late if there is this asymmetry and first excitation and only later inhibition. This inhibition will come too late and there will be an output. But this was an idea. It was an idea that there is some asymmetry in location of inhibition versus excitation, and in one direction first inhibition comes in and veto the excitation that comes later. In the other direction, first excitation comes in, and when the inhibition is active, it's too late. It was an idea. So how do you solve this idea? Very good idea, but what do you need to do? Basically, you need to see whether the anatomy that I just mentioned as a possible anatomy really exists in the system, and that's where the connectomy comes in. So we already mentioned in the first lesson that there are these new techniques, and there is a group of people using these very sophisticated techniques to cut and reconstruct fully the connectomics. Kevin Martin from Zurich was one of the first to try to connect anatomy at the level of synapses and neurons to a particular computation. Winfried Denk, who developed the two-photon microscope and now doing all this connectomic work. Jeff Lichtman from Harvard, Sebastian Sung from MIT, Mitya Shlovsky from Janelia Farm, Clay Reed now in Allen Institute. All these people are trying to reconstruct at the level of synapses, because I need to know where these inhibitory synapses sit, where these excitatory synapses sit, when are they activated. So I need both anatomy and electrophysiology to connect the two in order to understand whether the computation that I suggest is really implemented, can be implemented by this particular system, yes or no. And really recently, we have a breakthrough in trying now to reconstruct both the orientation selectivity of a particular cell and then reconstructing the anatomy surrounding the cell, the environment of the cell, at the level of synapses using this connectomic approach. And I'm not going to go into the details now, just to explain to you that eventually this work, and this is coming from Winfried Denk work from Heidelberg showing, and his team showing that indeed, if you look at a ganglion cell, this is the green cell, this is the directional selective cell that was really recorded from. And also, you look at the presynaptic cell, the amacrine cells, making inhibitory synapses onto the ganglion cell. So this is the amacrine, which is an inhibitory cell. And you can now count the inhibitory synapses. Here is one inhibitory synapse between this presynaptic cell to the ganglion cell. Another one, another one, another one, another one. Now you can count all the synapses. You can see the asymmetry immediately. You can see that to the right of the green cell, you get more inhibition than to the left. If you just count how many synaptic inputs there are, inhibitory inputs, you see there are more red inputs here and less red inputs there. Okay? This will be the source of direction selectivity, because now you understand that if a point of light is moving this direction, from left to right, it will first activate less inhibition onto this cell. Then excitation will come to this cell and will tend to activate it. If the input is coming from right to left, you will activate more inhibition. Then the excitation will be vetoed by this inhibition. So this could be, it's not completely resolved yet, but this could be a source from this directional selectivity, but now grounded in real experimental work. Both characterization of this directional selectivity physiologically, but also anatomically charting, drawing the environment of synapses that this cell is receiving. This is only the inhibitory synapses. So this is a breakthrough because now we can connect the anatomy to function, from structure to function. Let me summarize by a personal note this lecture. I want to say that the brain differs from other physical systems in many ways, but in one way in which the elementary units are in other systems, in most physical systems, the elementary units are simple and uniform. And you cannot really count the effect of one atom or one molecule on the overall physical system. But the brain is composed of nerve cells, of neurons, which are inherently complex, as I showed you. And they are dynamic and they are plastic. And I think this eventually, the formation of a network built in this case from complicated units, makes this physical system differently than the classical physical system where you can take an average of many, many elements, each one is simple. Here there are many, many elements, but each one is complex and each one may implement specific computation. So as in with humans, I want to say metaphorically that the individual neuron seems to enrich the dynamic of the society. So each one of us is complex, and this complexity together generate a complex environment. You cannot ignore the complexity of the neuron if you want to understand society. Okay, it enables us, the neuron, the synapses, enables us to learn, to change, and eventually successfully functioning in this complicated, unexplained, unpredictable environment. So what we really need to develop, and it's still missing in a grand scale, we need to develop conceptual and theoretical tools for connecting the low level, so to speak, the neuron, the synapse, the low level of computation and learning capabilities performed by single neurons and their synapses to the global computational function of the nervous system. This is the central role of the field called computation neuroscience, is to go from the computation performed by synapses, neurons, as I just mentioned today, to the global computational capabilities of a large computing networks of neurons in the brain. And that's really the mission of computation neuroscience, which is a relatively new field, maybe 20, 25 years old. And finally, as I just tried to show you in the last example, only very tight interaction between theoretical ideas, modeling and simulations, and experiments, now with connectomics and other methods to record the activity of many neurons, only the interaction between theory and modern experimental tools and experiments will provide the very, very missing still breakthrough in understanding how does neuron utilize, how does the nervous system utilize the neurons and the synapses to eventually enable me, for example, to stand and speak here, and for you now to listen and understand, move, go to the fridge, take a drink, go outside, and do all other things that you do computationally using neurons, synapses, and the network that they are built from. So thank you very much. This is the end of lesson 6. In the 7th lesson, we shall discuss the Blue Brain project and the Human Brain project. So let's meet next week.